draw out what the sarcomere actually looks like. And let's kind of review the overall structure of the muscle here. So we've got this muscle body. And if we were to look closer within this muscle body, we would see many different repeating units called muscle fascicles. So this is a single muscle fascicle. If we were to look even closer inside of muscle fascicles, we would see a single muscle fiber or a muscle cell. And then if we looked even closer into that muscle fiber, we would see a myofibril, a myofibril. Now, along the myofibril, we have several different repeating units, which we call sarcomeres. Okay. And so we're going to look at an individual sarcomere here. So I'm going to circle this little region. And that's exactly what we're going to look at a zoomed in view of and sort of describe the different features there. Let's do this in black. Okay. So here we're looking at a single sarcomere. And we want to remind ourselves that one myofibril equals many sarcomeres. Okay. So right along the center of the sarcomere, we have the connection of thick filaments. And I'm going to draw those here in red. Oops. So right along the center, we've got the connection of thick filaments. We'll come back and label that in a second. And then out towards the periphery, I'm going to label in blue the thin filaments. Let's do this first. So that's the center of our sarcomere. And here we've got the periphery of our sarcomere. So these are the thin filaments which are forming the outer periphery of our sarcomere. Okay. And as I mentioned before, this is forming this Z-like configuration. We call this the Z line. Okay. Now let's draw in here the actual myosin head. So the myosin have several different heads that are sticking up from these filaments. So we're going to draw those on in the midline. So these are the myosin heads, and they are actually going to come in contact with the actin filaments. Okay. And we're going to talk about the structure of these on a few slides down, but these are pretty much just differently shaped proteins. These are filamentous proteins that are responsible for the shortening of muscle. Okay, so let's label these out here. So this is myosin. And that is what we call the thick filament. And then this is actin. Oops. Which we call the thin filament. Okay. So let's start by describing the lines that we can appreciate. So right down the center where each of these myosin filaments are connected, we call this line the M line. Right, so this line here we call the M line, right down the center of the sarcomere. Now on either side where we have the connection of actin filaments or thin filaments, we call that the Z line. And as you can see, it does have this Z-like configuration. We're gonna call that, we're gonna call that the Z line. Okay, so we've got two Z lines. My Siri is picking up something. I don't know what you're picking up, okay. All right, so we've got another Z line here. And so we described a single sarcomere as the area that is bordered between two Z lines. So Z line to Z line, that region is a single sarcomere, okay? All right, let's look at the different bands. So the bands, if you recall, I mentioned were the different staining regions that we can see. And so we've got two bands and one zone. So we've got the I band, and the I band is the region that represents the staining pattern of only actin filaments, only thin filaments, okay? So there's no overlap here, there's only actin filaments. We call this region the I band. And as you would imagine, this is going to stain a little bit lighter because these are thinner proteins, right? It's gonna have a lighter staining pattern because of the fact that we've got thinner proteins here. Now along the center, as we can see, there is some significant overlap, right? The thin filaments are overlapping with the thick filaments. And so that region of overlap is gonna stain a little bit darker than the I band, right? We've got two proteins that are denser, which form that overlap. And that region we call the A band. Right, so all of this is considered the A band. Okay. Now, there's an area that's within the A band where there is no longer overlap. There's just myosin filaments, okay? And that region, just myosin, just thick filaments, we call the H zone. All righty, so we call this smaller region within the A band where there's no longer overlap. We call this region the H zone. Okay. All right. So I'm just going to make one detail here. Let's just describe this a little bit. So a single sarcomere, we'll describe, is bordered by two Z lines. Okay, so we have a clear definition of what that sarcomere is. So this is the structure of a myofibril, right? This is what's happening within a myofibril. We have many of these sarcomeres that are repeating one next to the other, next to the other, next to the other. And they're basically made of these two proteins that are parallel to each other, the thick filament and the thin filament. And in order for muscle to shorten, in order for muscle to contract, what's going to happen, and we'll speak about this a lot later on when we continue looking at muscle the actual contraction of muscle is the thick filament is going to come in contact with the thin filament and drag the thin filament closer towards the M line. So the entire sarcomere is going to shorten as these two Z lines come closer and closer together. And when this happens on a subcellular level, the entire sarcomere will shorten, the entire myofibril will shorten, the entire muscle fiber or muscle cell will shorten, the entire muscle fascicle will shorten, and consequently, the entire muscle will shorten. And that is how we bring about muscle contraction. And that is why understanding the subcellular structure, right, this sarcomere structure, is so crucial to understanding how muscle contracts.